Hello, everyone. Good morning. I want to welcome you to the EMBRC Technology Webinar series. This is a space to inspire collaboration and cutting-edge research. While people are still joining, I would like to share a little introduction about these webinars and our organization. Well, with these webinars, EMBRC wants to promote knowledge transfer and innovation. We are very excited to bring together experts, researchers, and enthusiasts to explore the latest technologies and challenges in emerging fields like, for example, marine observation, marine aquaculture, marine biotechnology. We want to warmly welcome people who are following this webinar from Europe, but also from other continents. We have people coming from Asia, from America, and from Africa. Thank you for joining us besides the different time in your different countries. So these webinars will be presented by pairs. And each series will include a speaker working in a research lab from academia and a company sharing its expertise. For those who don't know very well our EMBRC community, I will briefly present our organization. You can find the link in the chat. So the European Marine Biological Resource Center is a research infrastructure that provides access to marine organisms, ecosystems, data, technology platforms, and expertise for scientific and applied research. We support a wide variety of users coming from the private sector and from academia, touching topics such as environmental monitoring, marine biotechnology, marine conservation and ecology, developmental biology and evolution, drug discovery, and many others. Through our research infrastructure, we connect more than 80 marine stations, universities, and research institutes established in 10 countries in Europe. With this huge network, you can imagine that there is a very wide range of applications that EMBRC supports. So the topics of our technology webinars will be changing over the time. Our first webinar series will be dedicated to marine aquaculture, which is a field in which Europe is a leader in research and development, and for which EMBRC has facilities for performing, for example, feed trials, microbiome studies, environmental impact assessments, test innovative disease control strategies, genetic studies applied to selective breeding. We can also provide expertise in side stream valorization, in upscaling, and many others. All these services, I encourage you to access using our transnational access program, which allows you to travel and visit the facilities to perform your experiments, to perform proofs of concept, but also discuss with experts in the technology of your choice. So very soon, we will be opening our transnational access program in an EU project that is called AquaServe. You will find the link again in the chat. AquaServe is a joint venture of 6 research infrastructures who will offer access to research services for sustainable aquaculture and fisheries. Some of these services will be present in today's webinar. So stay tuned for submitting your application soon. Well, now that we have all of our participants online, I will introduce our speaker of the day. So Vincent Vermeylen, he's representing the Belgium node of EMBRC. Vincent is a biologist and scientific staff of the Ghent University's Laboratory of Aquaculture and Artemia Reference Center. So we have the link of Vincent's lab in the chat too. His work focuses in aquaculture research, and he's involved in studies related to larviculture, live feed, and aquatic health. He's also a member of scientific international committees, like, for example, for the Larvae Conference 2024, which was a great success, and I congratulate you. He contributes to discussions in broodstock management, in larval nutrition, and disease prevention in aquaculture. Vincent also loves to spread knowledge and awareness on aquaculture, so you can often find him at scientific outreach events. So today, Vincent will be presenting 3 themes on applied research, all focusing in sustainable aquaculture practices at Ghent University. I invite all participants to write your question in the Q&A section, And please type your name and the organization you belong so we can enable you to speak at the end of the webinar so we can all have an interesting discussion with Vincent. Welcome, Vincent, and the floor is yours.
Good morning, Mary. Thank you very much for the introduction. Thank you very much for inviting us to this series of webinars and being the first webinar in this series. So yeah, Mary already introduced me very well. So I suggest that we go into the presentation where I will also briefly introduce our lab, and then I'll go into detail on the sustainable aquaculture practices that we do here at Ghent University. So let me see, I can start sharing. Okay, just to double check. And I think we are ready to go. You can still hear me well?
Yes, everything's fine.
Okay. So, good morning, everyone, again. So, this is the first technology webinar from EMBRC, where we focus on sustainable aquaculture practices at Ghent University. As Mary told before, my name is Vincent Vermeylen. I'm scientific researcher at the Laboratory of Aquaculture and Artemia Reference Center, which is based at Ghent University in Belgium. So, more specifically, we are part of the Faculty of Bioscience Engineering, Department of Animal Sciences and Aquatic Ecology. You can find us here on the left. But besides our lab, I have to mention another very important lab when it comes to aquaculture at Ghent University, and that is the research group of phycology. They are part of the Faculty of Sciences and the Department of Biology, and they are very, very valuable partners when it comes to aquaculture research. You will see throughout this webinar, there is a lot of research going on seaweeds, on marine macroalgae, and that is all credited to them. So throughout the webinar, you'll see when it comes to aquaculture, animal science, it's mostly our side. When it's aquaculture on the seaweeds, it's from their side, but sometimes that overlaps in some projects. Short introduction on what we do in general in our labs. So at the Artemia Reference Center, so our lab, we have three main pillars. First of all, we have our research, of course, which covers 4 main research lines. We also support education, so we coordinate 2 master programs in aquaculture, and we provide, we have been providing for decades contract services to many partners, mostly the industry, but sometimes academic partners as well. The research lines in our lab, they focus on the first place on host pathogen interactions. So then I am talking about any form of microbial management, because the general topic in our lab is on larviculture, and microbial management is one of the key aspects when it comes to larviculture. We do this research on various animal models ranging from marine and freshwater fish to bivalves, shrimp, but also live feed such as Artemia. Which brings me to the live feed, which has been a core expertise in our lab since the beginning. Hence, we are named the Artemia Reference Center. And besides Artemia, there's also a lot of expertise when it comes to rotifer and macroalgae. Our third research line focuses on blue growth and sustainable aquaculture, which in fact means that we are looking for alternatives. Alternatives in the sense of alternative culture locations. For example, as you see in the picture, we are investigating the possibilities to grow low-trophic organisms at sea, both nearshore and offshore. On the other hand, we also look at alternative protein sources or feed ingredients, which can be Artemia or also insect larvae, or as you will see later, seaweeds. Besides that, this research line also supports nature restoration, which I will also explain later in the webinar. Our final research line is on the valorization of unused aquaculture biomass. And say this means that we recycle or we investigate recycling aquaculture waste and byproducts, can be from different industries, but also valorizing non-native species. As in many places, we sometimes are confronted with the issues regarding invasive or exotic species, and just removing them is one option, but reusing them, it's even more interesting. Our second pillar that I mentioned was the education. And here, in short, I just showed that we have two master programs that we coordinate. Maybe some of you have heard of it. The oldest one is our Master in Science in Aquaculture that has been running since 1991. It has been supported through VLIR and is actually our core program. Recently, or since a few years, we are also coordinating Erasmus Plus program, which is called Master of Science in Health Management in Aquaculture, which is together with partner universities in Barcelona, Wageningen, and Trondheim. Finally, our contract services in the lab cover different aspects. You can see here a few examples or the most common ones. It ranges from experimental design to live feed enrichment trials, feed trials. A lot of proximate analysis are requested. Live feed technique trainings are often organized, and flow cytometry can be one of the analysis as well. I placed the QR code here should you be interested to see what else we can offer for you. So feel free to visit it. And then, of course, our main partners, the Phycology Group. In short, they look both at fundamental and applied research in aquaculture, all on marine macroalgae. And they cover various topics ranging from genetics to diversity, growth development, life cycle control, one of the key aspects, especially when it comes to aquaculture, but also culture techniques, as you will see later on. So also here, there is a QR code. If you're interested to see what more they do, you will find their website using this way. Okay, so then we come to the main aspect of this webinar, which is the sustainable aquaculture practices that we do at university. So let me first introduce the three themes. First of all, we have marine multi-use and low-trophic mariculture, wherein say we look at the various culture techniques and monitoring techniques of low-trophic organisms such as bivalves and seaweed at sea, together with other marine activities. Our second theme is based on land-based aquaculture, and more specifically, we are looking a lot into the hatchery and the nursery phases of multiple species, but as you will see also, land-based grow out of seaweeds is a growing trend as well. And then finally, we have a smaller project where we investigated alternative feed ingredients and their impact on shrimp growth. And this was also together with our colleagues from the phycology team. So let's dive into the first theme, which is the low-trophic aquaculture and the marine multi-use. All right, it's a bit of a delay. So, as I said before, this is the culture of low-trophic organisms at sea. It can be nearshore, it can be offshore, and it focuses mainly, at least in our research groups, on local marine bivalves and macroalgae. So, for example, the blue mussel, the European flat oyster, and the brown sugar kelp. Multi-use, what is that exactly? Well, it's basically using the same area for multiple functions or functionalities. And specifically at the Belgian area, we have become very good at this. You can see here, there's a map of the Belgian part of the North Sea, and there's a lot of colors in there, and each color represents a different kind of activity. A lot of activities that need to be fit in this small area, so that requires a lot of planning, a lot of puzzling, a lot of lobbying, and of course, a lot of research. The green areas that you see here, they are marine protected areas, so that's a very important aspect to take care of. But then you also have the brown zones, which are the zones for renewable energy, and in between you see the overlap, where we even try to fit in aquaculture, as you will see later. So a lot of multi-use happening in this small area. So low-trophic aquaculture and multi-use at the Belgian Sea, specifically. In the recent years and currently, we are focusing a lot on bivalve culture, which can be in different techniques. But also seaweed culture is emerging a lot, as well as oyster reef restoration. Now, we do this not just on our own. I mean, that would be great if we could do that, but we need a lot of partners for that. And through our projects, for example, here you have a few examples of offshore and nearshore projects. You have the United and the Ultfarm project and the Idolis. They cover the growth or the culture techniques for bivalves and seaweeds at sea. Weir and Wind was specifically for seaweeds, and the MPV Aqua actually focused on the integration of bivalve culture together with a floating voltaic panel. And then we also have a few projects that cover restoration, can be specifically on oyster reef restoration, which is the Reef Recovery project. But then the Blue Connect project also covers any possible restorations in our North Sea area in general. Who do we do this with? We have national and international projects. For example, the VLAIO, FW, as the national funding agencies, but also Interreg and Horizon Europe are common projects around. We do this together with other academic partners. You can see here on the left, there are many. This is just of some of the few labs that are involved within Ghent University, each with their expertise, which ranges from aquaculture on our side to the Maritime Institute. We have the marine biology team sometimes involved, and so on. So besides that, we have a lot of private partners. Sometimes they are or partners that have affiliations with the government. So in general, you can see that there's a lot of expertise that comes together when it comes to making efficient use of our seas. Now, why exactly do we do this kind of research? I mean, yes, aquaculture is a valuable research line for food production, but we also contribute a lot to other societal and environmental targets. Low-trophic aquaculture, or the culture of low-trophic organisms, has a very low environmental impact compared to high-trophic organisms. For example, if you compare mussels and seaweeds to fish or shrimp, the mussels and the seaweeds, they require no input of feed, which means that there is less nutrients that go to the environment. That's why these low-trophic organisms also are often referred to as extractive aquaculture. Secondly, in many of these projects, there can be a very tight link with nature restoration and biodiversity improvement. Many of these activities sometimes also create like natural nurseries for other organisms. And specifically for our zone, as you will see in the next slides, the wind farms that we work in or that we collaborate with, they are assigned as safe zones, which means that there is no fishing allowed. There is few vessels that pass through, so only for maintenance or research, which means that any activities done there, especially those with a focus on nature restoration, biodiversity improvement, they are in the safe zone, so it means that they can develop very well without too much interference. Another important aspect is that we notice there is more and more incentives from government going on. So many projects require, whether it's for the private industry or for scientific research, they focus on low to zero, or in some cases even positive impact that has to be made on the natural environment. So making sure that biodiversity and ecosystem services are well maintained or even improved. So to highlight a bit of the expertise that both our labs cover, I have a few examples here. We have investigated a lot of culture methods for oysters at sea. So on the pictures here, you can see different types of baskets, different types of racks and nets that we investigated and that we placed at different locations, some near shore, some offshore. We also looked at different configurations of nests, so as you see in the right corner here, top right corner here. Sorry, lower right corner here, you see a different configuration of baskets within a frame. We also looked at bottom culture here in the middle. You see like a metal frame. And of course, also a lot of techniques to place oysters on ropes, as you can see on the right top culture. They were investigated. Some are better than others, but for that, feel free to ask us. We can tell you more about specific projects. The vessel that you see, they are from project partners. They're not from us. And you can also see that we also are faced with many challenges. Sometimes basket gets lost, so we need to redesign it. Sometimes we have a lot of fouling on our structures, which means the animals are difficult to find. So we learn a lot through these projects, and we have made a lot of progress. Besides that, we also have done a lot of research and currently are doing a lot of research on the remote setting of oyster larvae. So on the right top corner, you can see an example from an experimental setup where we investigated different substrates on which oyster larvae can settle. And the main idea behind this is that the scour protection around the wind farms, wind turbines, as you can see here in the middle, is made out of big rocks. And as I said before, it's a safe zone. So the idea is to like inoculate or seed these rocks with oyster larvae that hopefully turn into bigger oysters and eventually into small reefs, which then should boost the local biodiversity and ecosystem services, but also bring back the oyster reefs as we used to have them many decades ago. A few examples that we are proud of. You can see on the left, we looked at different substrates, including shells. Animals were growing very well. They were also grown in a closed and recirculating environment. On the right, you can see an example of oyster that were seeded onto the different substrates. On the left picture, you can see the tiny dots, which are the larvae a few weeks after they've settled. On the right, you can see the bigger oysters, which is around 2 months after they have settled. So you can already imagine that if you have a lot of these rocks with a lot of these oysters, you have small reefs that start growing. Culturing is one thing. You have to maintain it. You have to keep monitoring it. One of the most important aspects here is monitoring diseases, specifically in oysters. Most of our project, or actually all of our projects, including oysters, they focus on the European oyster, the Ostrea edulis. But unfortunately, there is also a parasite that sometimes can interfere. So we have to monitor for its presence, not just for the health of the animals, but also to avoid that we would introduce the parasite from one area to another. And we do this through histological screening, multiplex qPCR. You can see here an example which was made in collaboration with the Ifremer Institute. Okay. And then, of course, that was mostly on the oyster part. We have another low-trophic organism that's very interesting and relevant, and those are the microalgae. So you can see here on the left is my colleague. She is investigating different seeding methods on different types of nets, different types of substrates. And then they place these nets at sea, and then they monitor the growth of the macroalgae. Here's some example. You can see the small sporophytes that are seeded on the net. So they are like mixed in some kind of glue. You have to ask the details to them, to my colleagues, that they spray on the nets. These grow then into tiny sporophytes, which eventually grow into larger sporophytes. This is, of course, followed by well, the cultivation methods at the larger scale. So then we go at sea. You can see here some examples of the different nets that we put at sea. It's from different projects. And you can see here that most of the research is also done on the brown kelp, the sugar kelp. It's a brown macroalgae, Saccharina species. And you can see that it's growing very well in some conditions. It's not just growing, they also take samples for monitoring growth, as you can see here on the right. Samples are taken for genotyping. There's a lot of research going on behind the cultivation itself. Of course, as we had with the oysters, there's also many challenges that we face here. You can see, for example, on the left, you have different seeding methods that they used. You can see one is growing better than the other. Densities are very important, but also fouling, as you can see on the right, good timing can make a difference between a lot of fouling or almost no fouling, which impacts a lot the weight of the nets, and of course all labor that comes with it. So that was it this was a short overview of what we do when it comes to multi-use at sea and low-trophic mariculture. Our second theme, as I mentioned before, goes towards the land. And we looked into the nursery stages of different species, as well as the grow out of macroalgae. So what does it actually mean? So polyculture, it's very common, very old way of farming organisms. It's a very ecosystem-based approach. It's very sustainable. But in aquaculture specifically, it's rarely done at younger stages, like, for example, the hatchery and the nursery. So in these projects, we looked at requirements for young animals and young seaweeds in a RAS environment. So for those who are not familiar with these terms, RAS means recirculating aquaculture system, and it's in short actually a closed system where we aim to reuse as much of the water as possible. The focus here, as I said before, is on obtaining or producing starting material from three different species groups. We focus on bivalves, as you can see on the left. We focus also on crustaceans in the middle, and we focus also on seaweeds, as you can see on the right. The nursery stages of these organisms, they were investigated in the Blue Marine project. The grow out of the seaweed was done in a RAS environment as well, land-based, and which was first done in sea crops at a smaller scale, which went very well. And then we had a follow-up project, which is called the Cetamine project, where we are actually now upscaling the production or the cultivation of Palmaria species. So how do we tackle this? So of course, the main question is it feasible? But you need to have a lot of knowledge for that. So both at the single species and the multi-species level, we tackled many topics in these projects. I think one of the most important ones are broodstock management and life cycle control, rearing tools, feeding protocols, different types of feeds, all with a sustainable touch within the project. So again, who do we work with? Most of these projects were actually national projects where we looked at more local, regional level. We have a lot of academic partners that are involved. Here are just a few of them. We have a lot of private partners that are involved, all local, all Belgian, and they all are involved very closely to aquaculture. You have, for example, producers of oysters, of macroalgae, seaweeds, but also retailers are interested in what could be in this project for them. So you have a lot of collaboration with people that are in the industry sector. So why specifically, besides the idea of cultivating the younger stages on land? Well, there's a lot of issues when it comes to obtaining starting material. And by that I mean spat from bivalves, larvae from crustaceans, or seeds from seaweeds. There's a lot of issues when it's come to obtaining sufficient starting material, but also starting material of a high quality, because without a good quality, you cannot run your farm or your research. Secondly, there's a lot of issues when it comes to timing. Not all these high quality starting materials are available on the right time. Sometimes they are very dependent on the season. There's also a lot of requests from aquaculture initiative and research projects to obtain these starting materials. Especially in the last decade, you can see that there is like a boom in aquaculture initiatives, whether it's small research projects, European projects, small production initiatives, or bigger farms. Most of them, they source their starting material from another place. They don't produce it themselves. And the few suppliers around, they cannot supply everyone, all places at all time. And then lastly, what is also important is that biosecurity and environmental impact policies, they have become very, very strict in the last years, especially in Europe. We are very strict on this, which is a good thing, but it also makes some aspects of the research a bit difficult, especially when it comes to obtaining starting material. So that's why we have these projects running. More specifically, this would also support like a more broad supply of the starting material, and when you can grow these different organisms in a closed environment, you have very controlled conditions, which means you can improve the quality much more. You can produce locally, because you're less dependent on water sources. You can produce almost year-round if you have the right conditions controlled. There is even the possibility that you can produce disease-free or specific pathogen-free animals. And maybe you remember the slides where I talked about the European flat oyster, which has a lot of issues when it comes to Banamia parasites. Well, this makes it difficult to import them or to obtain them for research projects and for grow out. So this project would provide a solution for that when it comes to reintroduction and import. And finally, we also investigated what are the possibilities of IMTA, which is integrated multi-trophic aquaculture. It can be in a very broad sense. You can put animals or the seaweeds all together, or you can just recycle specific parts. It can be food leftovers, it can be waste, it can be water. Everything that contributes to reusing, recycling, and the materials. Okay, so again, a few examples of what we are doing here at our university. So we have a very good knowledge on RAS design. We make very modular experimental setups. They are very species specific. All of these are also homemade. So depending on the request of the project or specific requests, we can fine tune this. For these projects, there are a lot of challenges when it comes to designing a RAS for larvae or juveniles from shrimp or bivalves. And you can see here many aspects are involved. which also took us a lot of time to get to this point, but we made a lot of improvements in these projects. We also have a lot of rooms. We have like 7 of these rooms available, which means that we could run a lot of different trials at the same time. So you had trials running on the seaweeds, on the crustaceans, and on the oysters, which is very, very interesting because you can really screen better for synergies when everything is done in a relative short time span. So then specifically, our lab also has a lot of expertise when it comes to larviculture of crustaceans, specifically the Vanace vaname. I do have to give a side note, it's indeed not a local species, but it's a very good model species to fine-tune your experimental setup. So here, it was never done before, or at least rarely done before. It's rarely documented, but we managed to obtain a protocol to grow these shrimp from the mysis stage onward in a recirculating system. Mysis starts more or less on 7-8 days after they hatch, depends a bit on the conditions. So that's already a very good improvement. We have very good live feed production and control in our lab. As I said again, as I mentioned before, we are the Artemia Reference Center, so a lot of knowledge there. We produce microalgae, but we also collaborate a lot with other partners that can provide us high quality, high quantity microalgae of sometimes difficult to obtain species, which makes it very more interesting. Using flow cytometry, we could sample a lot of samples at the same time, or screen a lot of samples at the same time. And we use this, for example, to monitor the residual algae concentrations in the shrimp tanks of different species at the same time. And using that, we could fine-tune our feeding schedules and even reduce the amount of feed that we need. So very nice improvement. Besides the shrimps, we also have the oysters. You might have noticed before that we have a lot of research running on bivalves. Also just a few examples here. So we've done several settlement trials before. You can see a small setup where we let the larvae settle at on the oyster shells. On the left, you can see a small setup where we grew the bivalve spat, the oyster spat, in the recirculating system. We compared this with a batch system using similar conditions as they use in hatcheries, and we obtained very, very good results. So again, here a lot of improvements were made and a lot of skills were collected. Of course, our colleagues from the microalgae team have been busy as well. They have unraveled a lot of knowledge on life cycle control from many different and commercially relevant microalgae species. And for example, here you can see life cycle control of a porphyra species, where they unravel the different triggers that cause the different spores and different types of phytes, gametophytes, sporophytes that grow. And then they investigate more the different growth conditions to see which are better for the different cultivation methods. On the right, you can see also different recipients that they investigated, different culture methods for different species. It ranges from porphyra to ulva species to palmaria species. These seedlings, they are then used in the follow-up projects. For example, here again, porphyra, palmaria, and ulva, they are grown until the juvenile stage, as to say, so juvenile sporophytes, which they leave to grow on small scale in the lab. And then, especially for palmaria and ulva, they have developed larger recirculating systems where they grow them both indoors as well as outdoors. So they have a very good control on the culture conditions here. Most of these larger largest setups, they were built in collaboration with VLIS, which has the marine station in Ostend, and they have a very nice space and ideal location for this kind of experimental setups. We have very nice results here. You have good growth of these species, but if you want if you want more details, I suggest you contact the phycology team. And then this brings me to our last. This is a smaller topic. Sustainable and alternative feed ingredients, which also matches with one of our research line on alternative protein sources and so on. This was actually a follow-up from previous projects where we looked at integration of different species, but also reuse of resources. It can be something simple as reusing leftover seaweeds that are otherwise not fit or do not meet criteria for other conditions. So why not see if they can be used or reused? And here, one of the ideas was to use seaweeds as an alternative ingredient for aquafeed, specifically for shrimps. Why exactly will we do that? And not just to reuse it, but there have been, if you are working in aquaculture, then you're definitely familiar with the fact that animal-based ingredients are used a lot in aquafeed, and this is not a sustainable way of producing feed, or at least not for the longer term. So we are in need for other resources. And plant-based ingredients have been used a lot, but seaweeds not that much. So we tackled that topic. The culture of seaweeds, of course, comes with a low footprint, as I mentioned before. We focus also on local species and not always like the endemic ones. So we also looked at local invasive species because, as I said before, I mean, you can remove invasive species, but you can also reuse them. And one example here is Sargasso muticum, which has invaded some of the regions here at the Belgian coast, which is available and plentiful. So specifically, this was a close collaboration again with the phycology team. Their team has access to many resources from many species. These are just a few examples here that were used in this project. And you have a few brown algae, red algae, and green macroalgae. We looked both at invasive as well as native species. And for many species, not always these, but for many, they have culture collections and they have very good control of the life cycle and growth conditions. So very interesting, which means we have access to resources from, whether it's cultivated or not. And then this project, we continued by processing the seaweeds, the macroalgae into the feeds. We can do this all ourselves in the lab at a lower scale, but it's possible, it makes it convenient. Within this project, which was a student project, she looked into the antimicrobial effects also of different extracts of these macroalgae using minimum inhibitory concentration techniques and spectrophotometry. Very convenient and very fast. We did a nutritional characterization of these of the test feeds compared to the control pellets that we combined them with, as well as of the animals themselves. We also screened for their characterization. This approximate analysis is also done in-house. This is something that we do on a very, very frequent basis. So we have a lot of experience there as well, specifically for aquaculture, organisms, aquaculture samples, but also marine samples in general. Feeding trials are very common here, so it was also part of this project. So shrimps, they were divided over different tanks, they were given different diets, and then the student, she collected a lot of data, she did a lot of work, very nice work, where she looked at the nutritional composition of the body, she looked at different growth parameters, and then she also screened the microbiome of the gut of these shrimp after they were fed the different feeds. This is just one of the examples I want to share with you. So the gray bar is the control group. You can see that the protein composition in the pellets after they have been mixed with the algae. At 5%, it was more or less. You can see that some of those feeds, they don't affect the protein composition in general. So they are very interesting candidates. The microbiome screening was done by dissecting the midgut, and then DNA was extracted, was amplified, sequenced using nanopore sequencing. All the data, which was a huge data set, was cleaned and processed, and then they were analyzed for the composition and the diversity indices. So I have an example here again, where you can see the different treatments and their composition of the different microbial species. This was also done in close collaboration, this part specifically with the phycology group. They have a lot of experience when it comes to screening microbiome, not just for shrimp, but for seaweed and water samples and other samples as well. So we are very grateful to have such a very reliable partner at Ghent University. And that actually brings me to the end, because these were the three teams that I wanted to share with you. How can you contact us if you have questions, if you're interested for research, for collaboration, or whatever? This is the name. This is our location. We are located at Coupure Links in Ghent. This is our website, where you can find a lot of links to other activities. I have our email addresses here. We have a general address, which is artemia@ugent.be. There's my email address. You can find my QR code to my LinkedIn profile if you prefer to contact me that way. I will leave it a bit just in case someone wants to make a screenshot or check the links. And of course, our colleagues at Phycology team, they're also ready to help you in case you have questions. You can contact them through their website, phycologyugent.be, or through their general email address, or you can contact Professor Olivier de Klerk, who is the head, the director of this team. I also placed his QR code of his LinkedIn profile in case you like to connect with him this way. And that brings me to the end of this webinar. Thank you very much to everyone for listening. I hope it was interesting. I hope you can find some synergies with your research or your background. And I also like to thank the EMBRC team for inviting us and showcasing what we can do here at Ghent University.
Well, thank you very much, Vincent. It was a very nice presentation, and you gave a very nice overview of all the applied research that we can perform in UGent. I want to, maybe you can stop sharing your screen and we can go to answer questions. We only had one question from Antonella Petrocelli, who has been answered through the chat. What I would like maybe to know, if is there somebody else who wants to raise hand or ask any other question, please raise your hand or type it in the Q&A. And I have a couple of questions maybe in the meantime. So
Sure.
The outputs of the projects that you presented, as they are so applied and as you have also collaborations, well, some of your partners are from the private sector. What do they happen to these results? Are they absorbed directly by the different companies, or are they public, or how do you manage that?
In most of these projects, this is actually agreed upon in advance. So we know who can receive which information. Most of the times, this is accessible for all project partners. But in some cases, we have partners that also invest a little bit from their side. And then, of course, they use or they aim also to improve their activities throughout these projects. So it depends a little bit on the type of project and the type of knowledge that is collected. Yeah.
Okay, great. This is important because there is privacy that could be respected, even though people are collaborating with academia. I have another question, for example, regarding the multi-use systems that you have tested. Do you do some environmental monitoring where you actually measure the impacts in biodiversity of these different sets?
Yeah, that's done. It's not by us. We collaborate a lot with the Royal Belgian Institute for Natural Sciences. They are often involved in these kind of projects, and they are also members of the EMBRC. And they have a lot of knowledge when it comes to impact assessment in the environment, to biodiversity monitoring. They handle a lot of this kind of requests frequently, and their expertise is very, very valuable in these projects. So yeah, it's definitely done.
Very interesting. So we have some questions that are coming. So
Okay.
I don't know if Natalia Fitrillanti would like to open your mic and tell us about your question. So maybe while she's doing that, I will start reading the question. So Natalia wants to know, how sustainable is shrimp farming in Europe, and what is the biggest obstacle for shrimp farming?
All shrimp farming in Europe, as far as I know, is done in closed recirculating systems. So it's still developing, like there's still many challenges there. But the biggest one is actually to obtain sufficient and good quality starting material. So that's why I also mentioned it here today. That's one of the main challenges. When it comes to sustainability specifically, I think that there's a few cases where the farmers are really investigating every different aspect of closed recirculating systems. I know some of them that have managed to produce really or create like a nice closed shrimp farm where they recycle almost everything. So it's very intensive in terms of labor and time investment, but yes, it's doable, and it's very sustainable, or it can be very sustainable, I would say, formulate it like that.
Thank you very much. Okay, so I will go to the next question. So Sandra Ramos, thanks you for a very interesting presentation. So we are allowing Sandra to open her mic and share her question. Sandra, if you can open your mic.
Good morning. Can you listen to me?
Yes, if you can
Yes.
maybe say where are you from, which organization and which country are you from, and then you can ask your question, please.
I am from a collaboratory lab, Lab of Aquaculture in Portugal. And I'm working right now with macroalgae, but I'm using this macroalgae to feed sea urchins. And we are going to produce some pelleted diets. And that's why I put it this question, because I saw that you were using the macroalgae to include in the diets also for shrimps. And I wanted to know if you have tried lyophilizing this algae or you directly dry it, and which will be your recommendation?
No, I would have to look into the protocol from the student in our lab, but there was no lyophilization involved. It was more exploring different algae that we had available. But it's a very good suggestion. We had it in a discussion to see, as a follow-up for this. But maybe you can share me your email or so, or if you can contact me through my email, because this is a very interesting topic, and I'm sure we can discuss more about this.
Okay, I will do. Thank you very much.
Okay, thank you.
So thank you, Sandra. We'll go to the next question. So Georgios Kotoulas, he wants to know what is the origin of the industry you are contacting from, like what is the geographical origin? Georgios, if you can maybe introduce yourself, you can open your mic.
Okay, thank you very much for this great presentation, Vincent and Thank you, Mery Piña, for organizing this. This is a very nice, I liked so much the combination of, say, aquaculture part with ecological origin. It seems, I think, that the ecological origin came first. So it's very nice marriage, I think, and it's exemplary in my opinion. So I would like to ask, as you are, Belgium is a very small country. You presented it in the beginning. You don't expect an industry to have gains from the mass of production. I guess it's mostly if they expect to some say business, this would be from the part of selling technology or sell this kind of things. What does it comes mostly from Belgium, and why it would not come from elsewhere, for instance?
Well, in Belgium, as you saw, we are a small country. We don't have mass production of aquaculture, that's true. But what we do have, we have a lot of research going on here, and not just at the academic level, also at the private industry. There's a lot of research going on. And I think the major fields here are feed producers or feed additive producers, as well as companies that focus, for example, on pathogen screening, treatment alternatives. And that's something that we have worked a lot on. Some of these contacts with industry, they've also grown historically. So yeah, many are from Belgium, but we sometimes also have requests from outside Belgium. Although I have to say it's 99% it's from Europe, but it does remain within the field of mostly feed producers, looking for alternatives, looking for other ways to handle or to use or to apply their products, as well as like I said, disease monitoring, disease screening.
Nice, thank you very much.
You're welcome.
Thanks, Georgios. So we want to go next to Jessie Lubia. So she thanks you for the presentation, and she will ask you her question. Jessie.
Thank you so much, Vincent, for that wonderful.
Hi, Jessie.
Hello. Yeah, I was at the EMBRC for my master's. I graduated in 2021, but I just wanted you to highlight if there are any opportunities currently for PhD, if you have any scholarships or any partnerships that willing students can take up in order to complete their PhD studies at the lab. Thank you so much.
They sometimes they sometimes appear, and I have to say more and more because there's so many fields that are growing. But Jessie, I would say contact me. You have my email.
Okay, thank you so much. I'll get in touch.
You're welcome.
Thank you, Jessie. So next question is from Violen Dolfo. So Violen, if you can open your mic so you can ask your question. If you can maybe say where are you from or from which organization, that would be nice.
I cannot open my mic. I don't know if you can hear me.
Yes, we
I can hear you.
Oh, okay, cool. So thank you very much for this talk. I am from the Institute of Marine Research of Norway. I'm not working with aquaculture, so I just came as a curious researcher. And I had a question on the oyster reefs that you talked about, which are associated with wind farms. And I wanted to know if we have any data on the long-term monitoring of this reef, and if we know how much of the ecosystem and the trophic chain they can bring back, because I've seen a lot of starting studies, so but not much of long-term monitoring.
No. Well, but there is not much on long-term monitoring data. You say it yourself. Many of these pilots initiatives they've been running only in the last years. They're at small scale. But if you're really interested, I can contact you with a colleague that looked into the spread of oyster reefs and various environmental parameters connected with them. So that's a bit of the first rough long-term monitoring data we have, but it's not exactly on oyster reefs associated with wind farms, but it's a start.
Okay, yeah, thanks. That would be great.
So I think what we can do, Vincent, if you agree, is maybe in the YouTube channel where we will put this recording. We can add some of the publications from your lab or from the different projects that you have.
Yeah, sure.
Present. That's great. And our last question is from Natalia Fitrillanti. So Natalia, if you can open your mic and say where are you from or where are you based, in which company or university you work? You have to open your mic because we cannot hear you. I don't know if we can unmute Natalia from the back end.
Right.
Well, I will read her question. So how do companies in Europe, now Natalia, we can hear you now.
Sorry.
It's okay, we hear you.
No problem.
Yeah, I'm from Indonesia, by the way. And now I'm working in laboratory shrimp farming. Yeah. In Indonesia, the problem is EMNV, neonecrosis virus.
Mm-hmm.
Is the problem in my company. So how sorry, my English is not good, sorry.
Okay, Mery, can you maybe read the question if she already wrote it in the chat?
I will do that. So he is asking if shrimp are infected with diseases, it is still very minimal to overcome, and early harvesting is carried out.
Yeah.
Well, it's more on preventive measures. So, like I said before, most shrimp farms in Europe, also at a smaller scale, and not like the huge shrimp farms in Southeast Asia, but they work in very closed and controlled environments. That's one thing. And secondly, there is very tight control on the shrimps that come in. And maybe you remember that I said that it's very difficult to obtain high quantity, high quality starting material. It's also specifically for the shrimp. So it's difficult to get, but when we get it, we are very strict on screening them and using closed systems to maintain it should it happen.
Thank you, Vincent. I hope this was clear for Natalia. And we are coming to the end of the webinar. So I just want to thank everyone for their participation, for following us to the end. I want to thank Vincent very warmly for accepting this invitation. And I want to give special thanks to our EMBRC team, so our communications team, Gwenael Walter and Annabelle Chomum. And I will also like to thank very much my colleagues in the back end, Stella Alexandorf and Paulina Ramirez. Thank you very much for all your help. So this was the first webinar, and we are aiming for much more. Our next webinar will be still on aquaculture, and we will be now inviting a company to share with us their expertise. So stay tuned for the future, and thank you again for being here. Have a nice day.
Thank you. Have a nice day.
Bye.